Alright guys, so this is the video over gene regulation and cell differentiation. This is just a quick review over it. And to start off, let's talk about what cell differentiation is. Okay, it's just a process where stem cells, which are cells that are not specialized, become specialized, right? We talked about how stem cells are kind of like unemployed cells, and once they differentiate, then they become the cells that they're going to be for the rest of their lives, like heart cells or nerve cells or stomach cells, whatever, tissue cells, whatever cells they're going to become. That's what's called cell differentiation, going from stem cell to the cell that they're going to be, right? And there's a lot of factors that can make this happen, a lot of external factors, and we talked about how proteins regulate gene expression traits as well as sometimes cell differentiation. Okay, so that's what cell differentiation is. Okay, just becoming a more specialized cell, or stem cells becoming the cells that they're going to be. Okay, now the reason that heart cells and skin cells are different is because different sections of the DNA of that organism are being coded for that specific cell. We said that yes, the heart cells and in this picture, okay, the heart cells here, okay, are going to have the same DNA as the immune cells or white blood cells, but okay, maybe the heart cells are using genes 1 and 2 to make that type of cell, and then genes 1 and 3 are being used to make the immune cells. Okay, so that's how they become different. They're different because different parts of the DNA is used to make that specialized cell. So remember that. Okay, now they all have the same DNA, but different parts of the DNA is what codes for this specific cell. All right. Now we've completed the graphic organizer in class, and we said that there was 5 ways that gene regulation or gene expression, okay, it's regulated. Okay, and we said the first thing that can regulate what genes are expressed in an organism is the modification of DNA. Okay, and the way we modify DNA or our genome, okay, which is the entire makeup of the DNA in an organism, is by moving DNA around. Right? We talked about this, and we said this was a type of translocation mutation. Okay, if you have a DNA and a chromosome, and those chromosomes exchange information, so this section goes over here to this one, and this section comes to this one, then that's a way we can modify our DNA and make different proteins. Okay, also if we change, or if we tag our DNA to turn it on and off, and we can do that through DNA methylation, which is when we add little tags to our DNA to turn certain genes on and off. Or we can also modify our histones, which is the proteins that DNA actually attaches to. Okay, if histones are modified, then they get really tight to where DNA is turned off, and sections are not going to be transcribed. Okay. Now, when we modify DNA or modify histones, then we create our epigenome, which means above the genome. Okay, our genome is our entire makeup. The epigenome is what actually codes for proteins. Okay, and changes, things that can cause changes in our epigenetics, our environment, what we eat, what we drink, what we smoke, if you do any type of drugs, all those things can actually tag your DNA and turn it on and off different genes. Okay, so again, genome is the entire makeup of your genetic makeup, all the DNA. Epigenome is what is actually coding for proteins. Okay, all right. And there's a picture, we talked about this, and it shows how these little tags are actually, sorry about that, so these little tags are actually attaching to the histones, which are those little circles, or the methyl groups are attaching to DNA, and they're turning it on and off. Okay, again, the idea here is that we're modifying DNA. If we modify DNA, we're going to be modifying proteins as well. Okay, and that's how identical twins, as they get older, or why identical twins, as they get older, their DNA becomes a little different. Even though they're clones, their DNA is different because different genes are turned on and off depending on what they're exposed to. Okay. The second thing that can regulate gene expression is transcriptional regulation, and all that is transcription can be turned on and it can be turned off. okay? And I'm gonna go ahead and go to the thinking map. Okay, it can be turned on or off. And if transcription is off, then no mRNA is gonna be made. Remember that what we learned about DNA or protein synthesis, we said that DNA gets transcribed into mRNA, and then mRNA is gonna be translated to proteins, right? Well, if their transcription is off, then that means there's not going to be no mRNA made. So that's how your cells regulate this genes from expressing, by turning off the transcription process. Okay. Another way, or the third way that your gene expression can be regulated, is through post-transcriptional control. Okay. And what that means is that changes can still be made to the mRNA once it's transcribed from DNA. Okay, remember, not all DNA codes from mRNA. Some of it some of it gets transcribed into tRNA, which helps transport or transport. Okay, amino acids, and that happens during translation. Our RNA is also made from some of that DNA, and it helps in translation because our RNA is the ribosomal RNA, and we know that the ribosomes are the ones that actually make proteins. Okay, now what happens after mRNA is produced? Well, mRNA gets spliced, but before we go over mRNA splicing, we need to understand that genes are just segments of DNA that code for proteins, and then those genes are also cut into smaller pieces, which are called exons, the ones that are used to make proteins, and introns. Those are the ones that are actually removed from the DNA, from the mRNA, and are not used to make any of the proteins. Okay, now we did a small activity in class where I showed you and we talked about what mRNA splicing is. But all it is, okay, is if you think of this letters here, okay, and I'm gonna use, let me use another color. Let me use this one. So we have, let's say we have DNA here, okay, and DNA gets transcribed. We know that transcription, okay, transcription happens. Okay. And when transcription happens, then that DNA is going to turn into this mRNA. And we're going to call these words, or these letters that are here, we're going to call them mRNA. Okay. But remember, post-transcriptional regulation can happen. So what's going to happen is these letters that are in blue are going to be taken out. And we said that the words or the sections of DNA that get taken out, in this case the X's, those are called introns. Okay, those are not used to make any proteins. And the other ones that are left, like these ones right here, the ones that are in circle, and we use our key circle equals exons. Okay, exons are actually put together again. And if you put this together, they make the word protein. And those are the ones that are actually are used to make proteins. And this process is called mRNA splicing, okay? Because we're cutting splicing. We're cutting mRNA into smaller pieces. We're taking out the introns. We're leaving the exons in there. Once that the exons are together, then they can get translated outside of the nucleus. Translation, and it happens at the ribosomes. Okay, ribosomes, and polypeptides are made, and we know that all these amino acids are going to be connected, and when the amino acids are connected, we know that they make a polypeptide, or in other words, they make a protein. So that's mRNA splicing, and it's just taking out the introns, leaving the exons in there, and coding for what we know as proteins or polypeptides. Okay, again, exons were the letters that made protein. Introns were the other ones that made no sense and they were taken out. Okay, now this is another example. Okay, again, you see the DNA here. This section right here is actually what's going to code for the protein. So transcription happened and only the exons and introns are left, and you got mRNA. But we got to take out, or your cells need to take out the introns, and that's when mRNA splicing happens, and then only exons are left and you can rearrange them in what, or your cells rearrange them in whatever order they need. Okay. All right. So let's keep going. The number 4, translational regulation. Again, that's the same thing. Okay. You can either have translation turned on or off. Okay. Proteins. Proteins can also be modified. Once the DNA gets transcribed into mRNA, mRNA is spliced, and then that all the exons that are left are translated into proteins. The proteins can still be modified, and the way they're modified is by the way they're folded. Okay, so proteins get folded after they're synthesized, and the changing the shape. Remember that. Structure equals function. So if this one, let's say that this protein is the protein for your nails, right? Or, hold up. Let's do something else. Let's see. This is for your skin. Okay. That's for your skin if it's folded that way. But if you fold it another way, like this one, okay, this could be the protein for your hair. Even though it's the same order, the same amino acids in the same order, just because it's folded different, now the protein has a different purpose. Just like enzymes. Enzymes are very specific. Enzymes are proteins. So again, the way they're folded, that's the specificity, or how specific something is going to be, okay? And it just has to do with the way they're folded. So again, this is the 5 steps of how gene expression is regulated. Remember that is regulated. It's not just going DNA to protein. There's a lot of things that can regulate what traits and what genes are being expressed. And remember that proteins regulate gene expression. Proteins control the traits, which are your physical characteristics, and they also control the cell function. Okay. All right. So that was a review over gene expression. Review your notes and be ready to go in class.